actually applied to proteomics. Okay, so how we use mass spectrometry to identify proteins, which is our overall goal. Okay. So if in proteomics we want to study the entire protein complement, what we want to do is identify what are those proteins. And so this just shows the workflow. So you start off with your protein, and then you digest it into peptides, usually with trypsin. And so you've got these peptides that are a suitable size to be able to analyse by mass spectrometry. And then if you want to identify the protein, you've got two methods available to you. One is peptide mass fingerprinting, and that's a fast method, but with lower confidence. Or you can do sequence tagging, which makes use of collision-induced dissociation, so tandem mass spectrometry, and that is slower, but it gives you higher confidence in your protein assignment. Okay, and we're going to look at both of those. Okay, so when you digest your protein with a protease, you get a set of peptides, and it's a unique set of peptides, a set of peptide masses that is characteristic of that protein. So if you measure those masses of the peptides as a whole, as a set, you can then identify what the protein is. So I've got an example here, protein sequence. Now, if we digest that with trypsin, we would, in theory, get all of those peptides, because those are the cleavages at the lysine and the arginine, which should, if we record a mass spectrum, give us something like this, where we've got each peptide observed in the mass spectrum. And you see, I mean, I've written theoretical mass spectrum. You see that they're all of the same abundance. But as we've been talking about in the previous sessions, well, this is not what you get. You get something more like this, okay? So you see different relative abundances in the mass spectra. And that's because some peptides will ionize more easily than others. And if they ionize more easily, then they're going to appear more abundant in the spectrum. So this is the point we were making about ion suppression. If you start off with one protein molecule, you're going to have one of each of those peptides, but you won't observe that in the mass spectrum. You'll see the ones that ionize more easily. But it doesn't really matter for proteomics, because you don't have to see every single one to be able to identify the protein. You just need to be able to see a few. Okay, so if you're using peptide mass fingerprinting, then the mass of the peptide is going to be the sum of the amino acid residues and any modifications. Okay, so if we take this example here, this peptide here, pentapeptide. Okay, so we've got the mass of glycine, methionine, alanine, leucine, and lysine. Those are the amino acid residue masses. You need to remember that you've got termini on this peptide, so you need to add on the mass of the hydrogen that's at the N-terminus and the mass of the OH group that's at the C-terminus. And then, of course, because it's mass spectrometry, it needs to be charged. You need to add on the mass of the proton to give you the charge. So 1 if it's singly charged, 2, of course, if it's doubly charged. Okay. Right, so this is an example of a real peptide mass fingerprint. This is from the protein BSA. So the protein's been digested with trypsin, analysed by mouldy time of flight. And so each peak in this spectrum corresponds to a peptide, proteolytic peptide, from BSA. Okay. So I said that this is quick, so that's one of the reasons why mouldy is quite often used in peptide mass fingerprinting. So you can put it on the mouldy slide, you can get a mass spectrum very quickly, and you've got this set of peaks. So the next stage then is to take these masses and do a database search. Okay, so the database search works by taking the protein database, and I mean, since genomes have been sequenced, we've got databases of all the possible proteins that could exist for a particular genome. And then the algorithm will take all of those proteins and do an in silico digest, so work out what would the tryptic peptides be if we looked at these proteins. And then it takes those theoretical masses of the peptide and compares them with what you've actually got in your spectrum. Okay, and when it matches a number of peaks, it says, okay, this is the protein that we've got. Okay, so this is an important point. The peptide mass fingerprint is going to depend on the protease used. Okay, so trypsin, as we said, cleaves at lysine and arginine. You might be using lyc, which only cleaves at lysine, or gluc, which cleaves at aspartic acid and glutamic acid. Okay, so if you had this protein, what would the theoretical mass spectrum be for a gluc digest and a trypsin digest? I'll give you a minute to think about that. I'm going to hand these out, and they might be useful now, but they'll be more useful in later exercises. You might have to share some. I'll just pass them on and pass them back. So I've only printed 30 off. Okay, so if we start off with the gluc digest then, so that cleaves at aspartic acid and glutamic acid. If you take that protein, then the first one will start at the N-terminus. That's going to be peptide number 1. Okay, so those would be the first 4. You get that? Yes? So if you were going to work out the masses of those fingerprints, or the masses of those peptides that form the fingerprint, you would take the amino acid residues, which is on that sheet that's been passed around, plus the N-terminus and the C-terminus and whatever the charge is, generally singly charged if it's mouldy. Okay, so then the trypsin would give you I had someone say, yes, I've just done the first 4. There are more peptides, I just haven't written them all up. Okay, so what you can see is that you're going to get different masses depending on which protease you use. Okay, so that's really important. For those of you that are doing the tutorial this afternoon when we're doing the database searching, that's a really important parameter. You have to make sure you get it right or you won't identify your protein. Okay. So I've alluded to this already. What are the drawbacks of peptide mass fingerprinting? I mean, the advantage is that it's quick, but the drawbacks are that you need a higher number of peptides. You need to be able to detect a greater number of peptides to be able to confidently assign the protein. And plus, you don't get any sequence information. So let's say you had those 2 peptides, okay, you wouldn't be able to distinguish between them in a peptide mass fingerprint because they've got the same overall mass. They've got the same amino acids but in a different sequence, and so they've got the same mass, they appear at the same place in the mass spectrum, you won't be able to tell the difference, okay, so that would confuse the database search. And so any hit that was returned would be less, you could be less confident in. Okay, so if we want to have greater confidence, then we would do sequence tagging. Okay. And the first steps of the process are exactly the same as if we were doing peptide mass fingerprint. So you've got your protein, you digest it into peptides. So you've still got that information that you can get from the peptide mass fingerprint. You'll still know what the set of masses corresponding to your set of peptides will be. So you've still got that information, but you now do some collision-induced dissociation, so that's the tandem mass spectrometry, which will give you the sequence information. Okay? And then you can search the fragments that you observe following CID in a database search, and that will allow you to identify the protein. Okay. So if you go into the literature and look at this paper, you'll find that those researchers calculated that you only need a short length of amino acid sequence to be able to identify the protein. So if you know 6 consecutive amino acids, that should be sufficient to identify your protein. So you really don't need to get full sequence coverage. And by full sequence coverage, I mean see all of the peptides, all of those proteolytic peptides that came from the protein that you're trying to identify. You just need that short sequence. Obviously, the more that you get, the more confident you can be in the protein assignment. Okay, so this shows an overview of the workflow. those of you that are doing 9 will be coming for tutorial in the lab later, and we'll have a look at the instrument that we do this on. This example here is a QTOF. Okay, so you've got your protein, you've digested it, so you've got a mixture of peptides. They're separated by liquid chromatography, and then as they elute from the LC column, they enter the mass spectrometer. Okay. Now, so hopefully you'll remember from last time, and this is really important, if you're going to do liquid chromatography, you need to couple it with electrospray, okay, because you've got a continuous flow of peptides. So this is done with electrospray. So as the peptides elute from the column, they're electrosprayed into the mass spectrometer. And the first stage is to collect what's known as a survey scan. And that tells us what peptides are coming off the column at that time. And that's like a peptide mass fingerprint, but it's at a particular sort of retention time. So retention times from the LC column. Okay, so that gives us the masses of the intact peptides. Okay. And remember, when I was talking about intact peptides, I mean the proteolytic peptides, I don't mean the intact proteins. Okay, so we've got the mass of the intact peptides. And then that survey scan will show that a number of peptides are eluting. So could be 2, could be 10. But we have to make a decision, and the decision we make is that the 4 to 6 of those most abundant peaks are then fragmented in turn. Okay? So you've got the survey scan where you see what peptides are coming, and then the subsequent scans say, right, we'll take this first peptide and fragment it and see what the B and Y fragments are, and then we'll take the second peptide and fragment it, see what the B and Y fragments, and so on up to 6. Okay. And then another survey scan's done, because in that time, the elution profile will have changed slightly, so different peptides will be coming off the column. So there's another survey scan, okay, what peptides have we got at this retention time? And then we'll take each one of those in turn and fragment them, get the B and Y fragments. So in that way, even though we've got thousands of peptides in our mixture, we'll get MSMS, so tandem mass spec, spectra for each of those peptides. Does that make sense? Yeah, okay, hopefully. Right, so this is a real example. This was a band that was cut out from a 1D gel, okay, and then digested and subjected to this, we call it LC-MSMS, so that's liquid chromatography tandem mass spectrometry. Okay. Now this is the sequence of the protein that was ultimately identified, and the areas shown in red are the peptides that were seen in that analysis. So you can see there's a whole swathe of that protein that wasn't observed in the mass spectrum. But that's okay. We've got sufficient information from these few peptides that were fragmented, that we got sequence information for, to be able to identify it. Okay. So as an example, this peptide here, this is its fragmentation spectrum below here. Okay, this is actually an example that I used last week when we were talking about this. It's a real example. Okay. Right, and I showed this slide last week as well, just to remind you of the different structures of B and Y ions. So Y ions are like truncated peptides, okay, so they've got the same linear structure. They've got hydrogen at the N terminus, they've got OH at the C terminus, and they're charged. So very much like the intact peptide. Whereas the B ion has got this cyclic structure. Okay, so if you're calculating the mass to charge of a B ion, it's simply the sum of the amino acids plus 1 for the charge. Okay, you don't need to worry about the N terminus and the C terminus because they're lost in the cyclization. Okay, so we were saying, what's the problem with the peptide mass fingerprint? Because the problem is that we don't get sequence information. We get sequence information if we do the fragmentation, if we do the CID. So, for example, if you've got these two peptides here, which would appear identical in a peptide mass fingerprint, because they've got the same amino acids, so they've got the same intact mass, but their fragments, as you can see, are different. So the B2 fragment, which is cleavage between the second and third amino acid residues, okay, you can see that you get different masses and so on. So you would be able to tell these two apart if you did the sequence tagging experiment. Okay, does that make sense? Yes? Okay. Right. So, this is an exercise. This is what you needed that sheet that I was handing round for. Here's a mass spectrum. So this is the fragmentation spectrum, CID spectrum, of a peptide, which is triptych. So the mass of the intact peptide, or its mass to charge, I should say, is 835.4 Daltons. And the peptide comprises 8 amino acids. So from this, you should be able to work out what the sequence is. So I'm going to let you do that for a few minutes and see how you get on, and then we'll go through it. But you will need that sheet that I've handed round, which has got the masses of the amino acid residues on. Okay. Do you know what you're doing? Not this exercise. anybody think they've got it? Anybody questions? Okay, good.
I've got 8. So.
You've got 8. Okay, all right, good. Do you think nearly there? Anyone? Think they're nearly there? Okay, right, I'll go through it. So I've had various comments. So we know that the peptide comprises 8 amino acids. Okay. And we know that the peptide is triptych. So what that tells us is that the C-terminal residue must be lysine or arginine, okay, because that's where trypsin cleaves. So we know that. We don't know which one yet, but it must be one of those. Okay. Right. So now you see in the spectrum it's got Y fragments and B fragments. So my advice is always use the Y fragments where you can, and that's because the Y fragment has the same structure as the intact peptide. So we can just, it makes subtracting easy because we don't have to worry about the fact that there's differences in the structure because they're exactly the same. We can just use the Y fragments if you can. So now remember that the Y fragments contain the C terminus. So if we were looking at the Y7 fragment, that's that one. Okay? Yeah? So if we want to work out what's in the first position here, we want to look at the mass difference between the intact precursor, which is 835.4, and Y7, which is 748.4. Is that right? Yeah, okay. So if you subtract the mass of the Y7 from the intact precursor, that will correspond to an amino acid residue on your sheet, which is serine, yeah. Okay. And then we can move along to the next amino acid residue. So here, we're looking at the mass difference between the Y7 and the Y6, so that'll tell us what's here. What's up?
No, that's not correct.
Doing it the wrong way?
No. It's the opposite to what I did, but it seems to end differently, so I was listening to you as well.
Okay. Right, yes. So a common error is to get the sequence the wrong way round because you've forgotten that the Y fragment contains the C terminus, okay, when you're using the Y fragments. Right, so what goes here then? What residue have we got here? Yeah. Okay, and then we keep going. Okay, so now we're at 5. So what have we got here? Yeah. So here, just subtract Y4 from Y5, which will give you somebody other than you. Come on, shout out somebody. Yeah. And we just keep on going. Okay, so next is asparagine. Has everybody got that? Yes, okay. And then Y2, and that is glycine. And then here, Right. And then, who's got phenylalanine here? Right. Yes, so, okay, it's a common mistake. Right. The Y1 fragment, the structure of it, is the amino acid residue, plus the hydrogen at the N-terminus, and the OH, and the charge. Yeah? So the mass of that Y1 includes these components. Okay. Right. So it doesn't matter when we're doing this subtracting, because those masses are just being sort of wiped out. But it does matter when you get to Y1. So I think it's 147, isn't it? Yeah, so it's 147.1. So you need to take away the mass of the hydrogen and the OH and the H, which is 19. Okay, and that'll give you 128 and tell you it's lysine. Yeah? Okay. Right, so given that we now know what the sequence of that peptide is, which of these three proteins have we got? Who said that? Protein C. Does everyone else agree? It is protein C, you're right. Relax. Do you want to say why it's protein C?
Because in the second one, you've got SB, G.
Yeah. So it's this peptide here. Okay. So because we calculated or deduced what the sequence of this peptide was, we can then look at the protein sequences and say, where do we see that sequence? And identify the protein. So we know we've got protein C, not protein A or protein B. Okay. So that exercise that we've just done is exactly what the protein database does, but it does it for thousands of peptides. But it's the same idea. It calculates what the sequence is and then compares the sequence with the protein. Okay, right, so I've got another one, which is a little bit more complicated. Give you a few minutes to have a go at that one, and we'll go through it. Can I rub this one out? Has everybody got this? Yeah? I'm going to go through it because there is a couple of things that I want to point out in it. Right, so again, it's a triptych peptide, so we know this is lysine or arginine. But we're going to be troubled this time because we haven't got the full run of Y fragments. But we start off with the Y fragments. Always use the Y fragments where you can. Okay, so we've got the Y5, which is this one. Okay, and so to calculate this residue, we take the intact mass of the peptide and subtract the mass of the Y5, and that should give you threonine. Does everybody get that? Yeah. Okay, and then we move along. Okay, so Y4, subtract Y4 from Y5, and that should give you serine. Yeah. And we move along again. Okay, and that should give you proline. Everybody got so far? Right, okay, so this is the first bit to be aware of. So we have got the Y2 fragment. And if you subtract Y2 from Y3, you get 113, okay, which is what? But it could be isoleucine or it could be leucine, okay, and we don't know the difference. There's isomers, and mass spectrometry can't, not just on the basis of mass anyway, tell the difference between two isomers. So we don't know. It could be isoleucine or it could be leucine. So we have to put both here. Okay, it's one or the other. Right, and then we're missing a Y1. So now we've got a problem. We're going to have to go and use this B5 fragment. Okay. So this is where the differences in structure will come into play. So the B fragment is the sum of the amino acids plus the charge. And the intact is the sum of the amino acids plus the N-terminus plus the C-terminus plus the charge. Now the charges cancel each other out, but if you're going to subtract the B5 from the intact, you need to take into account the N-terminus and the C-terminus. Okay, so you've got, what is it? So the 18 is from the hydrogen at the N-terminus and the OH at the C-terminus. And that should give you 128.1. Yeah, and that tells you that we've got lysine here. Okay, but we still don't know what this one is. So what I would do is take the Y2, which we know is it has the unknown amino acid, lysine, the C-terminus and the N-terminus, and the charge. Okay, so we want, is it 218? So that's Y2. The lysine residue. Now that is the charge plus the N-terminus plus the C-terminus. And that should give you 71, which is alanine. Yeah. Did you follow that? Yeah.
Is there a way you could work out the last one without actually using the B5 peak? That's...
Well, you could do, I mean, you know that it's lysine and arginine. So what you could do is trial and error. You know, if I take off the mass of lysine, is there anything possible? Or if I take off the mass of arginine, is there anything possible? If there's only one possible, then you could do it. But if, yeah.
I think I worked out the B5 or other differently. I just used the B5 and added up all the first four amino acids.
Yeah. That would work. That would work, yeah. That would work. Yeah. Okay. Everyone happy with these? Right, so it is 10:02, so that's time to stop. Now, who is, we started this session with this, who is doing this as M9? It's not so many of you then. So there's only a few of you expecting a tutorial now. Can you put your hands up again if you're M9? Put your hands right up so I can tell who you are. So it's 17 of you. Okay. Because it's a lab visit, we can't have 17 people all in the lab at once. So if I could have 2 6s and a 5, Steph, you can probably sit it out because you know what you've seen it all before. Right, so that leaves us with 16. Right, so that will be 2 5s and a 6 then. Now I'm going to ask you to organise yourselves, and I ask this every year, so please be the year where it all works perfectly. So if some of you could come up now at 11 to the 5th floor, and then some of you come up about 20 past, and some of you come up, or 15 to 20 past, and some of you come up about 30 past. That would be good, so that we can get everybody safely in the lab to have a look. Okay? I'll let you organise yourself, and I'll see the first lot of you on the 5th floor in 8 minutes.
Are you around